Hi students, glad you're back for part 2. And in this part of the chapter, we'll be talking about the electrical changes that happen to the nerve. So first we'll talk about what is going on with the nerve at rest, and that is known as your resting membrane potential. And then we'll talk about if that nerve is stimulated, it could go through a graded potential followed by an action potential. So we'll talk about all of these changes in this section of the chapter. First off, let's go through a couple of things and kind of agree on a couple of issues here. That when you have a positive ion and a negative ion, those attract. So these electrical forces attract, while similar forces repel. So if you have two positive ions, that would lead to repulsion, or two negative ions, those would repel from each other as well. Okay, another thing to keep in mind is that the bigger the force, okay, the bigger the force of attraction. So you have this electrical force right here is between these two ions only. So the force of attraction is smaller than when you'd have all of these ions in play. Okay, and it's also the force of attraction depends also on the distance between the ions. So the distance here is shorter, so the force of attraction is larger than what you see up here, where the distance is larger, hence leading to a less force of attraction. So how is it that we are able to measure the membrane potential in a nerve? So remember that the inside of a cell is negative, while the outside of the cell is positive. The outside of the cell is your extracellular fluid, and that holds true for your nerves as well. We do that using a voltmeter, where we will put this reference electrode in the extracellular fluid, while the recording electrode would be found inside of the neuron. Okay, so the inside of the cell is more negative compared to the extracellular fluid. And that is what we name the resting membrane potential. So the electricity across the membrane at rest. So this nerve has nothing going on. It is not stimulated or inhibited. It is at rest, so RMP, or resting membrane potential. At rest, the membrane potential is usually at negative 70 millivolts. Okay, it is a range, but on average, and most neurons at rest are at negative 70 millivolts. What plays into that potential? So there are a lot of ions inside of the cell. There are also proteins which are negative. While on the outside in the extracellular fluid, there are also lots of ions, some positive and some negative. The two that we are going to be focusing on are sodium and potassium. So sodium, don't worry about the exact values. I'm showing these numbers for comparison. But on the outside, in the extracellular fluid, we have a lot more sodium than we do intracellularly. When you compare that to potassium, inside of the cell, there is a lot more potassium than there is outside of the cell. Okay. Chloride that is negative is a lot more on the outside than it is on the inside. Okay. Well, you might be telling yourself, well, now you're confusing me because you're saying that the inside of the cell is negative compared to the outside, yet there is a lot of potassium ions which are positive. Well, the fact is that there's so many different other ions and proteins that play into this. So remember, inside of the cell, we have lots of potassium, which is positive, lots of proteins, which are negatively charged. We also have phosphates and bicarbs. So there are so many different things that play into this, and we are kind of, simplifying the process. And the resting membrane potential takes all of that into consideration. But the two that have the bigger effects are sodium and potassium. Number 1, potassium, followed by number 2, sodium. So this is how we measure that potential, the equilibrium potential, which basically means that if you put a solution that is high in potassium, two different solutions with a membrane in the middle, and that membrane is permeable only to potassium, when will we reach an equilibrium? And that would be at negative 90 millivolts. Don't worry about how this process works. What you do want to worry about is these numbers. So the equilibrium potential for potassium is negative 90 millivolts. And that again means that if you have that membrane, how that is permeable only to potassium, when is that cell going to reach a state of equilibrium where no more potassium is going in either direction? And that would happen when we reach a negative 90 millivolts. While the equilibrium potential for sodium, so again, if that membrane only allows for the passage of sodium, that would be at positive 60 millivolts. We already said that the resting membrane potential in the cell is negative 70. So if you look at it, it is much more closer to the equilibrium potential of potassium than it is to sodium, which means that potassium levels has a bigger role than sodium levels do when it comes to the resting membrane potential in nerves. So how do we develop that resting membrane potential? Again, we have those ions that play a role. Each of these ions, sodium and potassium, have what are known as leak channels, meaning that potassium is always leaking out of the cell and sodium is always leaking into the cell. So how does the cell take care of that leak? We have a pump known as the sodium-potassium ATPase pump. It is an active pump, so it is a pump that needs energy, generating the energy through the ATP using an enzyme known as an ATPase enzyme. Hence the name sodium-potassium ATPase pump. That pump is always pumping potassium back into the cell and pushing sodium back out where it belongs. Okay, so again, we have the leak channels, we have the sodium-potassium pump that is always taking care of bringing things back to normal. Now, which one do you think is more permeable or has more leak channels? Is it the sodium or potassium? It is actually the potassium. So remember again that potassium plays a bigger role in this whole process. So I'm going to read the sentence and then explain it in the next slide. So potassium is highly permeable, which translates into having more leak channels, but has a small electrochemical gradient. Sodium has low permeability, so has less leak channels, but has a larger electrochemical gradient. And again, I will explain that in this slide right here. So let's see, what are we looking at? This is your cell body. That's the axon, the neuron. And this blue surrounding is your extracellular fluid. The inside of the cell is negative 70 millivolts, higher potassium levels on the inside than we do on the outside. The inside of the cell is negative. Potassium is positive. So it would want to stay in the cell, okay? But the levels of potassium inside of the cell are higher than the outside. So that force would be wanting to push it out of the cell. So you have these two different forces that are in play. You have your concentration gradient and the electrical gradient. Concentration gradients would want to push it out through those leak channels, while the electrical gradient wants to keep it in the cell. And that is why we say that the potassium has high permeability, is highly permeable because of those leak channels, but it has a small electrochemical gradient. That negative inside of the cell will want to pull the potassium back in, okay? Which is the opposite of what happens in sodium. So sodium doesn't have a lot of leak channels, but the inside of the cell is negative, while the sodium is positive. So that force is going to want to pull more sodium into the cell. So sodium is pulled in through two different forces, the concentration gradient and the electrical gradient. So make sure you are comfortable with these terms. Knowing potassium has more leak channels, knowing that sodium both is pulled into the cell through both the concentration gradient and electrical gradient, while in the case of potassium, the electrical gradient is pushing it into the cell, while the concentration gradient wants to pull it or to push potassium out of the cell. So your resting membrane potential, again, it's usually a range, but our average is at negative 70 millivolts, and that's the number that we're going to be using. And the changes in all the potentials are due to the movement of the ions back and forth, and what corrects that movement, again, is your sodium-potassium pump. And as I said, because we have sodium, potassium, chloride, protein, keep on going, all kind of contributing to your resting membrane potential, that is... it is measured or it is analyzed using this equation known as the Goldman-Hodgkin-Katz equation. Don't worry about the equation, just know the name. Okay, but it takes into account the permeabilities of all of these individual ions. Okay, so it's an expanded version of what we did here for individual ions, but puts them all into this huge equation that we're not going to worry about how to do that. Okay, worry about the name and what it does. So a little terminology that we want to go over before we talk about the action potential. We have the term resting membrane potential, that's at rest, negative 70 millivolts, and we already talked about that. We want to know what depolarization, repolarization, and hyperpolarization are. So I'm going to use the graph to explain that. So this dotted red line is the resting potential at negative 70 millivolts. If the potential changes in the positive direction, meaning that it goes closer to zero, that is known as depolarization. If we overshoot above that zero level, so the membrane potential is now at zero, and then it overshoots above zero, so now we're in the positive territory, so positive 10, 20, positive 30, and so on. That is known as an overshoot. Throughout actually the textbook and the slides, depolarization and overshoot are kind of gathered together into depolarization. Okay, so we're really not using that term overshoot a lot. We kind of gather them both together into depolarization. So depolarization would be above that negative 70, and includes while you're in the positive territory as well. Now going back down to the resting membrane potential is known as repolarization. Okay, while if you overshoot below or less than the negative 70, so for example, down to negative 90 millivolts, that is known as hyperpolarization. Keep in mind that if you want to excite the nerve, it has to be depolarized. If you hyperpolarize a nerve, that means that you have inhibited that nerve. So for example, when you want to numb a nerve, okay, so for anesthetics, they hyperpolarize nerves. Now, really important are graded potentials. So an action potential is when a nerve is going to propagate or take that message down, take that electricity down to wherever it needs to go. A graded potential are electrical changes. They're very local that happen either at a receptor or a pacemaker or at a synapse. So they're very local receptor potential changes, and they may or may not become a full-blown action potential. So I'll explain as we go, and hopefully that'll make a lot more sense as we go through these slides. So graded potentials are changes in your membrane potential that are confined to a small part of the plasma membrane. So they are localized, they may spread or they may not. If they do spread, that means that it switched from a graded potential to an action potential. There is a threshold. So if the membrane potential changes from -70 to -55, that is your threshold. Above that -55, the nerve will go through an action potential. Anything below -55, that means we are still in this graded potential area. Graded means that the potential can change from -70 to -65 or -60 and so on. So there are different levels of change that can happen in the potential in that across that cell membrane. So these are graded. They are local changes, so they are not going to propagate along the axon. And again, we need these graded potentials in areas where the nerve or the receptor has to initiate the electricity on its own. So for example, a pacemaker. So the pacemaker of the heart is known as the SA node. The pacemaker has the ability to make its own electricity. Okay, it's kind of like a battery in the heart that leads to these heartbeats. Okay, so the pacemaker has the potential or has the ability to make electrical changes on its own. So that is known as a pacemaker potential. The receptors receive different kinds of information, so pain, touch, vision, hearing, and so on. Again, when they receive these signals, electrical changes have to happen in order to take these signals to the brain. These local changes that happen at receptors are known as receptor potential. A synapse, as we know from the first portion of this chapter, is where a nerve meets something else. Okay, and in the area of the synapse, there is the ability to make electricity pretty much out of nothing. Okay, and that ability is known as synaptic potential. Here you can see what happens on a local level in order to for a graded potential to occur. So you can see here the chemical stimulus that opened up this chemically gated sodium channel. So remember we talked about these different receptors with the ion gated channels in chapter 5. So this is an example of this happening. So this chemically gated ion channel opened, which is usually a sodium channel, opens up and allows for the entrance of sodium into the cell. That leads to an area of depolarization. So now the positive sodium ions go in, it changes the electricity on the inside of the cell. If we do not reach the -55, then this will remain as a localized graded potential. Okay, so again, it's all about how much sodium entered the cell. If the amount of sodium entered changed the potential on the inside of the cell from -70 to -55, that will lead to a full action potential. We have fully stimulated that nerve. But if the ion channels or not enough sodium entered the cell and the inside of the cell did not change to -55, that means that any change in the membrane potential will remain local and will not propagate down that. Now here we can see on this graph different things that apply to your graded potentials. So if you apply a stimulus, that led to depolarization, led to opening of sodium ion channels. Again, it did not reach the threshold, but it did cause local changes within that nerve. While other stimuli might open other kinds of ion channels, leading to hyperpolarization, which means that we've inhibited that part of the nerve. Okay, we can also see that graded potentials can be graded. Okay, so we have different levels of graded potential depending on the strength of the stimulus. So a weaker stimulus would lead to a weaker graded potential, and the stronger would lead to a stronger one. So again, there are different grades to that, and we could add them up to get a even bigger grade of a response. Okay, so know that these apply to your graded potentials, not to your action potentials. Also, depending on where you record the change. Okay, so let's go back to this image. So if you record the electrical changes that are happening here, it will give you a bigger curve. As opposed to recording it at this point, you will get a smaller curve. And that's what you see here. So measured at the site of the stimulus gives you a bigger curve compared to the curve you get if you've measured, 1 millimeter away from the stimulus. Again, all of this applies to graded, not action potentials. Now here you can see that the action potentials follow the all or none rule, which is extremely important, meaning that if an action potential will happen, it will happen at the same levels, so there is no grading. As you see in the graded potential, they are all at the same level. They either happen or they do not happen. So it's an all or none rule. So how is it that our bodies have the ability to differentiate between different stimuli, the strength of the stimulus? The strength of our stimuli does not depend on the levels of these graphs or these curves. It depends on the frequency of the action potential. Okay, so your graded potentials, depend on the strength of the stimuli. You have these different levels. Once you've reached a threshold stimulus, okay, you are going to have the same level of action potential even if that stimulus keeps on increasing. Okay, so how do we interpret the different strengths? We interpret them by the change in the frequency of action potential. So an action potential are large changes in the membrane potential. You have to reach that threshold of -55, and then it'll keep on depolarizing, and it can go all the way up to maybe even positive 100 millivolts. They are very fast. They could happen as 1 to 4 milliseconds. And they can repeat at very high frequencies, maybe about 100 per second. So again, depending on that frequency, that's how when you ask a patient, like on a scale from 0 to 10, how painful is this? Okay, so that level of pain really depends on the frequency of action potentials. The ability to generate an action potential is known as an excitability. And again, you have to reach that threshold. The propagation of action potentials down the axon is how the nervous system communicates with the other cells. So we'll see the different ion channels that the nerves have. So action potentials and graded really depend on different ion channels. Graded potentials depend on those ligand-gated and mechanically gated sodium channels. Action potentials do depend on those, but they also depend on the opening of the voltage-gated ion channels. If you do not open these voltage-gated ion channels, an action potential will not occur. So the graded potentials, again, are produced due to the opening of the ligand-gated or mechanically gated ion channels, which leads to the influx of sodium into the cell. If you reach -55, so you've changed the electricity from -70 to -55, that will open a different kind of ion channel. And because it opens due to the difference in voltage, these are known as voltage-gated ion channels. These voltage-gated ion channels, some are for sodium, other are for potassium. Okay, and those are the ones that we are going to be focusing on. So in your text, you are going to find that there are calcium, chloride, ions, and so on. We are going to be focusing on your sodium and potassium voltage-gated ion channels. And there's a little bit of difference between these two. So both of these are voltage-gated channels. The one on top is your sodium, the one on the bottom is potassium. Sodium ion channels are usually closed until, again, we reach the threshold of -55, and then they are open. And then they open up. So you see here, we have opened the sodium ion channels. Lots of sodium will rush into the cell. And when it reaches a certain level, usually at about +30, these sodium channels become deactivated. So you see that kind of, cork kind of shuts down or deactivates the sodium channel. So the channel is still open, but it's blocked. Okay, we call that deactivation of the sodium channel. So you see that your sodium channels can open and become deactivated very rapidly. If you compare that to your potassium channels, though, so potassium channels are closed. They will open up at +30 millivolts, but they do not have a plug. Okay, it'll take them a very long time to go back and shut down. So these open and close much slower than the sodium ion channels. Now let's take a look at the curve of the action potential. So right here is your -70 millivolts. Again, during this phase, this is where the graded potential occurs. So if we reach, and this is where your mechanical or chemically ligand-gated sodium channels are opening, if the inside of the nerve changes into -55, that means you have reached your threshold potential. At that point, this is where sodium ion channels open up, leading to a rush of sodium into the cell, leading to the phase of depolarization. At about +30 millivolts, okay, you see here that your sodium ion channels become deactivated, while the potassium channels open up. So potassium will rush out of the cell, leading to the phase of repolarization. Remember, we just said in the previous slide that potassium channels open and shut slower than sodium channels do. And because of that, potassium will keep on leaking out of the cell, leading to this hyperpolarization area. Remember, everything will go back to normal, so the cell will eventually go back to its resting membrane potential, where the sodium-potassium pump will pump the sodium back out and the potassium back in. So for these phases, you want to know which channels are open and which channels are closed. So why on earth do we need to know all of this about the action potential? Well, it is really important clinically because when we generate an action potential, we have stimulated a nerve. But on the other hand, we can also inhibit nerves during using local anesthetics. So for example, Procaine or Novocaine, lidocaine, also known as xylocaine, these are local anesthetics and they work by blocking the voltage-gated sodium channels. So even if a graded potential reaches the threshold, these anesthetics block or prevent the voltage-gated sodium channels from opening. So no matter how strong that stimulus is, these voltage-gated sodium channels are pretty much paralyzed and an action potential will not happen. Okay, so that person is not going to feel the pain. We also have some toxins, poisons that are made sometimes by different animals. So for example, a pufferfish, okay, that is used in making a delicacy known as fugu, that has an organ that produces a toxin known as tetrodotoxin, and these do the same things, or that toxin does the same effect as local anesthetics. It blocks the voltage-gated sodium channels, okay, and prevents an action potential from happening. So why is this considered a poison while this is considered a local anesthetic? Well, this will produce the changes in your sensory nerves. This will produce the change in your motor nerves. And that would be a problem because if you block the motor nerves from working, especially the respiratory muscles, that could lead to death due to respiratory failure. So if you ever, want to eat one of these dishes, you want to make sure that it is by a licensed cook. Now for refractory periods. Refractory literally means stubborn, okay? So during these refractory periods, you keep on stimulating the nerve, but the nerve is so stubborn that they don't want to respond. So there are reasons why this happened and advantages to this. We have two different kinds of stubbornness, two different kinds of refractory periods. One is an absolute and one is relative. So during the absolute refractory period, the nerve will not respond no matter how strong that stimulus is. Okay, so there is complete stubbornness on the side of the nerve. So a second stimulus, no matter how strong, will not produce a second action potential. That again is known as your absolute refractory period. So we should know at this point that in order for an action potential to happen, we have to open up voltage-gated sodium channels. If the voltage-gated sodium channels are already opened or are deactivated, then the nerve has no ability to respond to that stronger stimulus. So I will show you using the curve where this happens. So the absolute refractory period happens if all of your sodium channels are already open or they are deactivated. So it occurs during depolarization and repolarization. Meaning that if the nerve is in the process of de or repolarizing, even if you stimulate it with the biggest stimulus possible, it is not going to respond to that stimulus. Okay, so again, absolute refractory period. But, okay, maybe I should stay on the curve. But a relative refractory period can happen in this phase right here. So where we have now this stage of hyperpolarization, where the voltage-gated sodium channels are now shut down. If a stronger than usual stimulus comes, we can open up these ion channels, leading to a second action potential. Okay, that would be your relative refractory period. So we would need a stimulus stronger than normal to lead to another action potential. Okay, so in your relative refractory period, a second action potential can be produced only if the stimulus is considerably greater than usual. And that coincides with your period of after hyperpolarization. Okay. So we'll see, what that looks like kind of on this graph. So this right here corresponds to the strength of the different stimuli, and this looks at the curves that they might produce. So you see here that threshold stimulus led to a very normal blue action potential. Now right here during the absolute refractory period, this stronger than normal stimulus, so you see here that is much stronger than normal, was applied, but the nerve did not respond. So you do not see a corresponding curve to this. While this red stimulus is also stronger than normal, it happened during the relative refractory period, during the hyperpolarization area, and it led to an action potential. Okay, so again, no matter how strong the stimulus is, if you're in absolute refractory period, no response occurs, and that occurs during your de- and repolarizing parts of the curve. And that again is because either all of the sodium channels are already open, so you have nothing left to open, or they are all deactivated. During the hyperpolarization area, you do have the sodium channels that can open, but you need a stronger stimulus to open them. That again is your relative refractory period. So what is the importance of these refractory periods? Well, first off, they limit the number of action potentials that a nerve can carry, which is really important. Okay, so you do kind of want to have some kind of filter to filter out these action potentials. And don't get me wrong, it's not like they're not fast. Okay, so they could carry up to about 100 action potentials per second, which is pretty fast, but you don't really need the overstimulation. It's also important in determining the direction of action potential propagation. And I'll explain that in this right here. So remember we talked about the propagation of the action potential away from the cell membrane on the axon, away from the cell body, down the axon. So the action potential will be going in this direction, but not towards the cell body. And that is, you want to thank the refractory period for that. So if you take a look at these three sections, A, B, and C, or 1, 2, 3, in area number 1, this is where the action potential will start, and then it will spread to area number 2. Area number 2 can either go back to 1 or go down to number 3. Well, area number 1 is in its absolute refractory period, okay? And therefore it will block the potential from propagating in this direction, and really we have no other choice but to go down away from the cell body. And so your refractory period helps in the direction of propagation. Myelination, myelinization helps in speeding up the process. So instead of going one step at a time in the propagation of the action potential, and that jump is known as the saltatory conduction, where it jumps from one bare area, to the next bare area. This bare area that is not covered with myelin sheath is known as the node of Ranvier. And again, we have these electrical changes jumping from one node of Ranvier to the next. Hence, the myelinization speeds up the conduction of the action potential. The thicker the myelination, the faster the action potential can propagate. Now, this table is extremely important. The differences between a graded and an action potential. So I'll go over it again one more time. So your graded potential, we do have different grades to it. We can sum them up. And there is no threshold, okay? So they are going to happen regardless. There is no refractory period. The amplitudes decrease with distance. Okay, so the further away you are from where the graded potential happened, the amplitude will decrease. The duration is going to vary depending on the stimulus. Okay, so a stronger stimulus will lead to a longer graded potential. A graded potential can either excite or inhibit a nerve. The graded potential is a local change that can happen either at the level of a receptor or at the level of a synapse or spontaneously, for example, in the pacemakers. The mechanism would depend on the opening of the ligand-gated ion channels. And these ligand-gated could either open up through chemicals or changes in mechanics. So they're ligand-gated and mechanically gated ion channels. If you contrast that with action potential, know that action potentials start off as graded, when they reach the thresholds, they are now an action potential. So an action potential is all or none. Once you've reached that threshold, you'll go through that whole curve. We cannot sum them up. Okay, so there is no summing up. We have to reach a threshold. There is a refractory period. Remember the absolute and relative. The conduction occurs without a gradual decrease in the level of action potential. Okay, so the duration is constant, so it'll go throughout down that whole nerve. It is only a depolarization. Okay, we cannot, an action potential does not mean that you have hyperpolarized the nerve. An action potential can only occur if you depolarize the nerve. As I said, they are all of these action potentials start off as graded potentials, and the mechanism depends on the voltage-gated sodium ion channels. When those open, you have reached the threshold, the whole curve will occur. So it really depends on the opening of the voltage-gated sodium ion channels. Those are your first ions to open. This is a mini glossary. We've talked about all of these different definitions. I am putting it out there because it is extremely important, the definitions. You'll also find them on the chapter outlines. You want to be familiar with these. If anybody has any questions, please let me know.